Hello everyone, this is Shinuja, Assistant Professor, Department of Biotechnology, St.Josephs College of Arts and Science for Women, Hosur. Today's topic is recombinant vaccine. Vaccine. A vaccine contains inactive or weakened section of a specific biological entity, that is antigen, that triggers the body immune system to provide immunity against infectious diseases. The entity stimulates the immune system that recognizes it as a foreign material and destroys it. This forms a memory in our cells, and in future, whenever that foreign body, that is antigen, invades our body, the immune system can fight against it. Recombinant vaccine. Recombinant vaccine are synthesized using yeast or bacterial cells for the manufacture of vaccines. These protein or DNA recombinants are introduced into the host cell where they are identified as a foreign material, and as a result an immune response is triggered. The advantages of recombinant vaccine is that they can be used for people with weakened immune system as well. However, one shortcoming is that you need to get booster shots to maintain the effects of the vaccine. Advantages. Advantages of the recombinant vaccine technology are that there is a virtually no chance of the host becoming ill from the agent, since it is just a single protein, not the organism itself. Traditional vaccine risks come from the organism not being totally weakened, that is, attenuated, or a reversion to a virulent, that is, disease causing form. Another advantage of a recombinant vaccine is that it does not need an adjuvant. An adjuvant is an agent that stimulates the immune system to find and react to the vaccine agent. Some adjuvants have been implicated in causing cancer in some animals over time. Different types of vaccine. The recombinant vaccines may be broadly categorized into three types. The first one is subunit recombinant vaccine. These are the components of the pathogenic organism. Subunit vaccine include proteins, peptides, and DNA. Attenuated recombinant vaccine. These are the generally modified pathogenic organism, that is bacteria or viruses, that are made non-pathogenic and used as vaccines. Next type is vector recombinant vaccines. These are the genetically modified viral vectors that can be used as vaccines against certain pathogens. Preparation. The basis of these new vaccine is, in the first place, the identification of the protein of the infectious agent that are able to induce an immune response in a similar way to that produced by the whole agent. Secondly, the identification of those proteins that are not immunogenic, do not have a role in replication, or that are related to virulence. Thus, these proteins are not necessary. Using genetic engineering, the genes coded for these proteins can be selected, cloned, and expressed using different vectors. They can also be eliminated by selective deletion. The procedure for insertion of foreign genes and vector can be grouped into four categories based on the construction and insertion of recombinant DNA. DNA source, selected vector, restriction enzyme, transformation into host. The source of DNA used to construct the recombinant DNA is usually a small part of a genome that contains the gene or sequence of interest. The gene or sequence of interest is usually of significant importance due to its unique function, whether it be the production of a protein or capsule or regulate gene expression. Once the DNA source is chosen, it must be isolated. This can be accomplished with the use of restriction endonucleases. The isolated DNA is then inserted into a vector. Vectors are used as a vehicle by which foreign RNA can be inserted into cells so that the recombinant DNA may be replicated. The most common type of vector is a plasmid. Plasmids are a small circular piece of extra chromosomal DNA found in bacterial cells, which are capable of replication independent of the host. Plasmids usually carry a few genes which give them a selective advantage, such as gene for antibiotic resistance. Next one is examples of recombinant vaccines. The examples of recombinant vaccines are human insulin. Next one is human growth hormone. Next one, human papilloma virus vaccine. Another one is BCG vaccine. Bacillic gurin is a vaccine for tuberculosis, that is TB diseases. Many foreign born persons have been BCG vaccinated.